Alright guys, welcome back to Underground Science, and in this video, we'll be going over the basic structures of a neuron. Alright, so this will be important to really get familiar with just the basic structures of a neuron so it can further help us in our nervous system playlist. Alright, so let's go and jump right in. So first, right, we're going to have these structures on our neuron. Alright, so actually, you know what, first let's start off by, let's see right here, let's just start off by just putting a really simple definition on a neuron. So what does a neuron do? So pretty much the very basic function of it, or just the overview function of it, is that a neuron just is responsible for mainly transmitting information, all right? Transmitting info, okay? And we'll see in other places too, like in the hypothalamus, we have a bunch of nuclei that actually release specific types of releasing hormones, like for example, adrenocorticotropic releasing hormone, right? Many chemicals that control the release of hormones in our pituitary gland, all right? But in general, just know that neurons are responsible for transmitting info, all right? So let's go and see a basic structure of a neuron. And to classify, like in the central nervous system, we have white matter and gray matter. So we'll make a video on the differences of white matter and gray matter, all right? So we can know the basic differences between white matter and gray matter, all right? But for now, let's just go and look at the structures, basic structures of a neuron. So let me go ahead and draw the soma first, all right? So the cell soma, or the neuron soma. So let me go and label this as the soma, all right? And so the soma is the same thing as the cell body, and in the soma we have DNA, right? So we have DNA inside our nucleus, all right? So let me go and draw that real quick. All right, we have DNA inside our nucleus, and then we have organelles also, but just to save time, let me just go ahead and draw the mitochondria, all right? Because the mitochondria is one beast, because it does a lot of important metabolic functions, as we'll see in the metabolism and cellular respiration playlist and all that. All right, so I'm just going to just try to draw the cristae here. All right, so we have that, right? This is the soma or cell body. All right, then we're going to have these structures that are going to be mainly responsible for receiving signals, all right, receiving electric graded potential signals. These structures are going to be called dendrites, all right? And dendrites are going to have tree-like branches, and these tree-like branches, if we zoom in on one dendrite, you're going to see many receptors on them. And those receptors are going to be responsible for receiving those signals, right? Let's say if a neurotransmitter binds or something, you could say on those receptors, either it's going to be excitatory or inhibitory, right? So it's going to overall, if it's excitatory, it's going to cause many depolarization, temporal summations, and graded potentials that involve spatial summations, right? So if you haven't checked out my graded potentials video, go ahead and do that, all right? Talked about temporal and spatial summation there. Or the signals that bind, so I don't know what these dendrites are, like that was a pretty bad drawing of it, sorry about that, but these are just know that dendrites are many long tree-like branches like these. And it's mainly responsible for, so we're going to write it down. So these are the dendrites of a neuron, and it's mainly responsible for receiving electric signals. All right, so let's just say receiving signals. Okay, signal/info, I should have said, but receiving signals is fine. All right, so we're going to receive the signals through here, through our dendrites. It's going to come through our soma, and it's going to meet at another place here, right before another structure. What is that place that we're going to, so what I should have said is that the signals are going to meet here, and in this particular place that we're about to talk about next is where the action potential is actually generated. So if all these graded potentials are big enough as it comes down to this particular structure will actually hit a threshold potential and will generate action potentials. Action potentials are nothing but electric signals traveling down an axon. All right, so you know what, let's just go and label the next structure. So the next structure we're going to talk about, it's obviously not drawn to scale, but what I'm trying to show here is that it's right before the axon. All right, so it's right about here, right before our axon starts. And this structure is called our axon hillock. All right, so our axon hillock is also going to be called our trigger zone. And the reason why it's called the trigger zone, guys, is that because in this particular structure, we see our action potentials being generated. So action potentials are generated here. All right. So the axon hillock is pretty much the deciding zone. All right. So the axon hillock will interpret all the graded potentials that are coming in based on how strong it is over here. If the membrane potential hits negative about 45 millivolts, then we'll fire action potentials. All right. Where will we fire action potentials? Well, this is our next structure right here. All right. So this structure that I'm drawing here is going to be called the axon. So let me go and color it out for you guys real quick. All right, there you go. So this structure right here that we just colored in, right, is going to be called our axon. All right, our axon is where the action potentials will travel through. So the electric signals that will travel through the axon will then synapse, right, and then we'll have neurotransmitter release, and that's how we'll communicate from a neuron to a target cell or a neuron to some other neuron. Okay, so let me just go ahead and draw action potentials going down, and we'll talk about more about action potentials in when we make action potential videos, right? The step-by-step physiology of what happens inside the neuron, and we'll talk about the steps of an action potential and what's happening at each specific step. All right, but those are the action potentials traveling down the axon. Then we reach a structure right here that's going to be called the synapse. All right, so it's going to be called the synapse. And so we have either the synaptic bulb here, you can call it, which involves the synapse, right? Or the axon terminal, right? So the axon terminal is actually what comes next, you could say, because it's where the axon ends, all right? So right here is going to be the axon terminal. Axon terminal, all right? And then we have our synapse right here, okay? So this will be our synaptic cleft. So let me go and draw let's say we're actually, you know what, let's just go and say we're trying to synapse onto another neuron. So let's say our dendrites of the other neuron is right here, and then let's say our cell body of the other neuron's right here, and then the neuron will continue that way, all right? So we'll actually have a synapse. So this space right here, guys, is going to be called the synaptic cleft, all right? And the synaptic cleft is where neurotransmitters go, all right? So this is where neurotransmitters are released from the synapse, all right? So actually in the synapse, we have specific vesicles. So if this was a lower motor neuron or something, then these vesicles would release acetylcholine, right? It's a type of neurotransmitter that's involved for muscle contractions, all right? And other things, but in particular muscle contractions, all right? If we're talking about the lower motor neuron. But anyway, so let's go ahead and review real quick, all right? So actually, you know what, we should label that this is the synapse, all right, or the synaptic bulb. Let's just label it to the synapse in general. All right, so let's go and review one more time. So we have the dendrites, and these dendrites are mainly responsible for receiving signals, all right, and transmitting those graded potentials through the soma and into the axon hillock, where we decide if we're going to fire action potentials or not. If action potentials are generated, they're going to travel down our axon. Then, once the action potentials reach the end of an axon, we call that the axon terminal. Then all of this is called a synapse, where we synapse onto another neuron or another target cell, all right? And this is where we release our neurotransmitters in the synaptic cleft so that we can communicate with the postsynaptic neuron or the postsynaptic target cell. So another thing is that this neuron right here, if this is the synapse, then this whole neuron is going to be called the presynaptic neuron. Presynaptic neuron, all right? And if this is another neuron, then I'm going to call this the postsynaptic neuron. The postsynaptic neuron. And the reason why it's called presynaptic neuron is because it's before, which is what pre means, the synapse, all right? So it's a neuron that's before the synapse, all right? Because the synapse is right here. This is called the postsynaptic neuron because it's a neuron that's after the synapse, all right? So it's postsynaptic. And we'll be talking again about the differences of white matter and gray matter in another video. But um, if we were to consider myelin here in this video, right, in this particular basic structures of neuron video, then the myelin, depending on if it's the central nervous system or the peripheral nervous system, right? So if you haven't already, go check my glial cell videos out. The glial cells of the central nervous system and the peripheral nervous system, right? There's more coming though. But anyway, if this was a central nervous system, we would have, and if this was a myelinated neuron, let's just say it was, then we would see myelin sheets around the axon if this is a myelinated neuron, right? And also, in particular, if this is the central nervous system, we're going to see these glial cells called the oligodendrocytes. So let's go and write that down. If we're talking about myelin, then let's classify what we're going to see in each of the types of nervous systems. So if we're talking about the central nervous system, we're going to have oligodendrocytes that are responsible for the production of myelin for the central nervous system neurons. So we're going to be talking about oligodendrocytes, all right, guys? And then if we're talking about the peripheral nervous system, all right, so if we're talking about the peripheral nervous system, then we're talking about a glial cell called swan shell, called a swan shell, right, or swan shells. Not swan shells, swan cells, all right, swan cells. And these Schwann cells are responsible for producing myelin for neurons that are in the peripheral nervous system. All right, so we're talking about neurons here. So neurons are also called nerve cells. All right, so the nerves in the peripheral nervous system. And we'll talk about the differences of white matter and gray matter in the central nervous system in another video. Okay, but that's pretty much it. That's what I wanted to get through in this video for the basic structures of a neuron. I hope this video made sense to you guys. I hope to see you guys in my future videos. We'll talk about more cool topics about biochem, physics, and chem. And so see you guys later. Make sure to hit that notification bell, like, and subscribe. And yeah, and stay safe.